Now we go to the third presenter, who is actually a Spaniard. She's actually doing research in Belgium. Maria Sanchiz Calvo will present exploring the role of the lysosomal lipid flippase ATP10B in the nigrostriatal dopaminergic pathway on rats.
Okay, good morning everyone. I'm Maria and I'm a PhD student in the group of Verle-Bakeland in Belgium. And today I'm going to try to bring you a bit closer to what we are doing in the lab. And the title of my presentation, as she said, is Exploring the Role of the Lysosomal Lipid Flippase ATP10B in the nigrostriatal dopaminergic pathway of rats. I don't have any conflict of interest to show. So in the recent years, and I think as you've seen through the whole Congress, and for example in the first presentation, the Parkinson research community is trying to find, or is doing tremendous effort trying to find new genes that are associated to the pathology of the Parkinson. In fact, in 2020, colleagues of our lab studying a Parkinson's disease and dementia Lewy bodies cohort in Belgium, they discovered new mutations in a gene named ATP10B, identifying this gene as a genetic risk factor, or as a novel genetic risk factor for Parkinson's. Interesting, these mutations are loss of function mutations, which means that they negatively affect the function of the protein that is encoded by this gene, which means that the protein is not functional anymore. But now you will be wondering, what's this protein actually doing? So ATP10B is a lipid flippase, which means that it uses the energy from the hydrolysis of the ATP to transport two specific lipids, phosphatidylcholine and glucosylceramide, from the inside of the membrane to the outside of the membrane. So as you can see in the picture, ATP10B, it's a membrane protein, and specifically, it's located in the lysosomes. As you may know, lysosomes are the garbage disposal system of the cells, which means that their main function is to degrade damaged protein and organelles that are in the cells. As for example, you can see in the picture, lysosomes are the main pathway where not only soluble alpha-synuclein, but also its aggregated form present in the neurons of the Parkinson patients are degraded. And also lysosomes have many other functions, like for example, functional connections with other organelles. And then is it not surprising to say that such a small organelle play a vital role in maintaining the cellular health. ATP10B seems to play an important role in these lysosomes because when researchers decrease the protein levels of ATP10B in the mammalian cells, they observed that there is an impairment of the lysosomal membrane integrity, as well as an impairment of the degradation activity of the lysosomes. So this brings to the next idea. As we have observed ATP10B loss of function mutations in the Parkinson's disease patients, what's the impact of the ATP10B loss of function in the Parkinson's disease neuropathology, if there is any? So if we go to the Parkinson's disease neuropathology, we can say that one of the main hallmarks is the impairment of the nigrostriatal system. And this means that in the brains of the Parkinson's patients, we find a loss of dopaminergic neurons in the substantia nigra, as well as a loss of dopaminergic terminus in the striatum, and as a decrease of dopamine release, which is one of the causes of many motor impairments that are observed in the patients. So if we rephrase the question a different way, our idea was if we decrease the protein expression of ATP10B in the dopaminergic neurons of the substantia nigra, are we able to replicate what is observed in the neuropathology of the brain of Parkinson's patients? To answer this question, for us as researchers, it's a bit difficult to work with the human brain because we are talking about the first steps in the pathology. So we actually work with Parkinson's animal model. In our case, or in my case, I'm working with the rat model. Why I choose the rat model? First, because the genome of the rats is similar enough to the genome of the humans, that they express the gene that I'm interested in, which is the ATP10B. And also because in both brains, so in the rat brain as well in the human brain, we can find the nigrostriatal pathway and the both regions that are implicated, which are the substantia nigra and the striatum. So as I said before, our idea was to decrease the protein expression or the protein levels of ATP10B in the dopaminergic neurons of the rat brain. And to do that, we knocked down ATP10B using viral vector-mediated shRNA. Probably these terms are a bit unfamiliar for some people, so I will try to explain in an easier way. In our study, we have 3 different groups, a control group, and 2 groups that I will call MIR5 and MIR7 from now on. What's happening in the MIR5 and MIR7 groups? So normally in the dopaminergic neurons, we have the messenger RNA for ATP10B that encodes for the protein. What we will do is to introduce 2 different sequences that will bind to this messenger RNA, and what they will do is degrade the messenger RNA and decrease the protein levels in these groups. In the control group, we will also introduce a sequence, but the sequence will not be able to bind to the messenger RNA, so the protein levels remain the same. We will introduce this sequence in the neurons of the substantia nigra using stereotactic injections in the brain and viral vector as a means of transport. So we will have these 3 groups with the 3 different sequences. We kept the rats alive for a year, and during this year we performed different behavioral tests to address different parameters of the motor behavior of the rats that actually can be comparable to some of the motor impairments observed in the patients, like the balance and motor coordination or muscular rigidity. We also performed PET imaging at different time points, and at the end we obtained the brains and we studied the pathology of the brain focusing on the dopaminergic neurons in the substantia nigra and the dopaminergic terminals in the striatum. I will show you now some examples of the behavior we perform in the rats. So this is what we call the open field. And in this behavior, we place the rats in a big arena, and we leave them move freely. And then we study different parameters of this spontaneous behavior. As you can see in the graphs, I show the velocity and the distance travel done by the rats. And I could observe that in both MIR7 and MIR5 group, which are in the blue and the pink lines, the velocity and the distance travel was decreased compared to the control. So I could conclude that when I reduce the protein levels of ATP10B in the substantia nigra, I have a less spontaneous locomotor behavior in the rats. This is another test, we call it rotarot. It's a moving rod where the rats, using their balance and their motor coordination, they need to keep walking on top of the rod without falling. And actually what we measure is the time that these rats are able to stay in top of these rods. And we can observe that in both MIR7 and MIR5 groups, in blue and pink in the graph, the time that they are able to stay on top of the rod is less compared to the control. So we can conclude that when we reduce the protein levels of ATP10B in the substantia nigra, we impair the motor coordination and balance of these rats. This is the last test that we're going to show, and we actually just performed it in the last month. In this test, we place the rats in an uncomfortable position, grabbing the bars. And as you will see in the control group in the up video, the rats normally come back to their normal position on the table. But what we observe in both MIR5 and MIR7 groups is that the time that the rats were in this uncomfortable position was longer. So we could conclude that when we reduce the protein levels in the substantia nigra of the rats, we observe an increased muscular rigidity in these animals. In order to track the progressive loss of dopaminergic terminals in the striatum, we also perform PET imaging. We actually follow a similar strategy as is done in the clinic sometimes for the patients. We use a tracer which is a PE2I, and this tracer binds to the dopamine transporter. The dopamine transporter is located in the dopaminergic terminals in the striatum, and we observed that the binding of this tracer to the dopamine transporter is lower in the MIR5 group compared to the control, and actually there is a progressive loss of this binding of the tracer to the dopamine transporter. So we could conclude that when we reduce the protein's level of ATP10B in the substantia nigra, we actually have a decreased presence of the dopamine transporter in the dopaminergic terminals. And this actually brings us the idea that what we actually have is a loss of dopaminergic terminals in the striatum. We could confirm this idea when we study the pathology in the brain of the rats. And when we observe the brain of the rat with this staining, we could see that we have a loss of the dopaminergic terminals in the striatum in both MIR5 and MIR7 compared to the control. And we also have a loss of dopaminergic neurons in the substantia nigra in both groups. So if we come back to the question that we were doing to ourselves before, so we observed these ATP10B loss of function mutations in the Parkinson patients. So we were wondering what's the actual impact that this loss of function of ATP10B could have in the neuropathology of Parkinson's disease. And using our model in which we decrease the protein levels of ATP10B to mimic what is happening in the mutation of the patients, we could observe that we have Parkinsonian motor deficits in our rat model. We also have a decrease of striatal dopamine transporter binding. And we have a decrease of dopaminergic terminals and dopaminergic neurons in both striatum and substantia nigra. So now the follow-up question for our study is what are the specific molecular mechanisms that are actually induced by this loss of ATP10B in the dopaminergic neurons of the substantia nigra pars compacta. I will just like to thank you all my lab, especially Edward is very involved. He's also part of this project and is working with me in the lab. A special thanks to ASAP, which is the main funding for the project, and we are part of a really big group, collaborative group of different labs, where Peter Vangelo is the leader. And if you have any questions about my research, I am also in the poster 06.48, and thank you very much.